Hello everyone, my name is Big Thuy Nguyen, and today I will talk about gap junction of cellular communication. So let's begin. Cellular communication has important role in multicellular organism by upkeeping homeostasis of tissue or organism. Using communication, cell can signal to adjacent cells in order to generate a coordinated response or a special groups of cells. Gap junction is one type of communication between cells formed by intercellular channels that cluster in specialized regions of the plasma membrane. The gap of the plasma membrane, usually between two cells, falls between 2 to 4 nanometers. Gap junction channels link the cytoplasm of two cells, allows the change of ions such as potassium and calcium, second messengers such as cyclic AMP, cyclic GMP, inositol 1,4,5-triphosphate, and small metabolites such as glucose between adjacent cells by electrical and by biochemical coupling. Gap junctions present in both vertebrates from Mesozoa to mammals. In higher plants, it is used in the form of plasmodesmata for direct intercellular communication. Gap junction channels are composed of connexin in chordate animals and characterized in three groups known as alpha, beta, and gamma according to their gene structures, overall gene homology, and specific sequence motif. In non-chordate animals, gap junction communication is conducted by a family of integral membrane proteins called annexins. In recent studies, another group of proteins known as pannexins also discovered show similar characteristics as annexin. But so far, only connexin genes shown to be linked to human disease. Each connexin is composed of 4 transmembrane domains with N and C termini and the cytoplasmic loop that linking the second and third transmembrane domains together. The domains are connected by 2 extracellular loops with 3 cysteine in each loop, forming an intra-connexin disulfide bonds that play roles in cell-to-cell recognition and docking process. The biosynthesis and assembly of gap junction are strictly regulated, and the intercellular junctions have a very short half-life with only a few hours. Connexins begin co-translationally into the ER membrane, and the oligomerization of 6 connexins make up a hemachannel occurs in a manually fashion starting in the ER and ending in the trans-Golgi. The hemichannels or the connexins are then carried to the cell surface by vesicles and get transported through microtubules and lastly get fused to the plasma membrane. Connexins play an important role in tissue or organ homeostasis, and mutation of connexins genes were linked to various human hereditary disease. One example is the erythrokeratodermia. Is a mutation of the CX31 or CX30.3. It is considered one of the isoform of connexins. Another known syndrome are known as the Clouston syndrome or the hydrotic ectodermal dysplasia. It's a mutation of the CX30 gene. And lastly, the palmoplantar keratoderma or focal hyperkeratosis and kinky hair. It is the result of the mutation of the CX43, also one of the connexin's isoform. The function of gap junction plays an important role in the body, and the nervous system is no different. Being able to measure and characterize the distribution, function, and regulation of gap junction, we have to understand why it has an important role in physiological and pathophysiological view. Different methods has been developed, such as invasiveness, signal-to-noise ratio, temporal resolution, in order to detect and characterize gap junction. The paper also go over recent developed chemical tracers that use chemical compounds, genetically encoded enzymes, transporters, channels, and fluorescence protein in order to map gap junction. Lastly, here's a question I got for you. There are 9 types of genes or isoforms of connexins. Do you know any other diseases that can caused by the other isoforms, or know someone who suffers from connexin gene mutation? Thank you for watching and have a good day.